Good afternoon, everyone. Thank you for this opportunity today. I am just introducing myself, Dr. Dinesh, co-founder deNOVO Biolabs. Today we will be talking about E. coli HCP detections, and Dr. Manjunath will be taking the sessions. So before that, I would like to just introduce about deNOVO Biolabs activities. We are a Bangalore-based company, works for antigens and antibody development for biopharmaceuticals and diagnostic applications. So we have been catering to this industry, biopharmaceuticals industry, from more than a decade now, and we have experience in developing these products and catering to biopharmaceutical companies, as well as providing them the custom services. So this session will go on, and at the end we will have a discussion Q&A sessions. So I would love to interact with each one of you, and we'll keep this session very simple and interactive. I would expect the participations from each one of you. Wherever you have any questions, any doubts, at the end of the sessions, we all can discuss together. So with this, I think we are set to go. Dr. Manjunath will start the presentation. Dr. Manjunath takes care of our R&D, and he is the key stakeholder for this R&D activities, and he will be taking through the E. coli HCP detection, what we have developed here in deNOVO Biolabs, and how it is a differentiating from the market available current solutions, what advantages we bring to the table and we bring to the scientists and regulators and to manufacturers. So I hand over the session to Dr. Manjunath.
Good afternoon, everyone. I'm sharing the screen, so hope it is visible to all of you. Dinesh, is the screen visible?
Yes. Yeah, okay. Screen is perfect.
Okay. So very warm welcome everyone for today's webinar, and thank you for joining today's session on detection of E. coli host cell proteins using ELISA. deNOVO Biolabs, as described by Dinesh, is a product development and customized solutions providing company to the sectors of biotechnology, biopharma, and biopharmaceuticals for the last 13 years now. I'm Dr. Manjunath. I take care of the R&D at deNOVO. In this webinar, we will cover what are HCPs and why HCP detection is critical, and how our solution of HCP detection by ELISA helps biopharma production and ensure the regulatory compliance also. During the process of production and purification of biopharma products from microorganisms like E. coli, mammalian cells, yeast, so some host proteins from the used microorganisms may still remain in the purified product, which is called as host cell proteins, or these are shortly called as HCPs. These residual HCP proteins can cause immunogenicity, reduce product safety, efficacy, and stability, thereby triggering the regulatory issues also. So monitoring of HCPs is not only optional, but an mandatory requirement in the biopharma sector. So out of many microorganisms used in biopharma production, E. coli is one of the most widely used microbial hosts. So however, during the downstream processing, it also brings with it a large impurity pool of its host proteins. So manufacturers and regulators require a sensitive and quantification of these impurities, and ELISA remains the gold standard method to detect the HCPs. Generally, here is how the sandwich ELISA principle for HCP detection works. We capture the anti-E. coli HCP specific antibodies on a microtiter plate and add the standard HCP standards and its test samples. So this complex is then detected with a conjugated anti-HCP polyclonal antibodies, and finally the signal is measured in a microplate spectrophotometer reader. The signal intensity of the standard samples is plotted, and test signals are extrapolated on it to determine the presence and absence of HCP in test samples, which directly reflects HCP amounts present in the test samples. So regulators expect a proof of assay sensitivity and protein coverage. They recommend validation like 2D gel electrophoresis and encourage process-specific HCP ELISAs for late-stage programs. So what options available for HCP detection currently are the imported commercially available kits, which are well established in the market. But the main factor is they are expensive and their supply is very delayed. So our indigenous developed HCP ELISA solution provides comparable sensitivity to the imported kits and perform a similar fashion. And they are more cost effective in price and faster product delivery. So being a local manufacturer, we could deliver the products quicker than the imported ones. Local technical support and the option to create custom kits are also available with us. So our HCP ELISA kit detects wide range of low and high molecular weight HCPs. The polyclonal antibodies are raised against a blended lysate from different E. coli strains, including the DH5 alpha, BL21, JM109, K12, MC1061. So what happens is this demonstrated adequate coverage and reactivity when we develop the antibody against these these all strains. So these process-specific assays may not be generally required on several occasions, but a custom assay may be developed to address this particular regulatory challenges. If anyone wants to use their own strain and we do not have the coverage towards those strains, so we could develop the custom assays towards the client's requirement. So we have developed and validated this HCP E. coli HCP kit as per the EMA guidelines. So the dynamic range, assay range, is around 500 to 2 nanogram per ml with a sensitivity of 1 nanogram per ml, which is quite comparable with the other commercially available products in the market. So the intra and inter batch precision were evaluated by spiking the E. coli standards at low, medium, and high QCs. The inter and inter batch percent CVs was observed to be less than 10 and less than 20% respectively, which satisfies the regulatory recommendations. So coming to custom HCP ELISA development, we can also generate the antibodies tailored to your unique processes and microbes used, ensuring better coverage and detection. So later the developed reagents can be converted into a kit format, and which can be made as an ready-to-use kit, wherein you can just add the standards and test samples and get the results in about 2 hours. So from immunization to assay validation, the timeline typically would be around 6 to 8 months to get to the assay. So to conclude and summarize, this HCP monitoring is really critical for biopharma products, and ELISA is the most reliable platform. And our E. coli HCP ELISA kit is sensitive, reproducible, customizable, and cost-effective, and which has a low turnaround time. We also offer the custom assay development for the clients who have the unique needs. Thank you for your time today. If you would like to more details, please reach out to us and contact us through email or call. And we really look forward to supporting your HCP analysis needs. With this, we are open to discussion with the attendees here, and we are happy to answer your questions. Thank you. Thank you again for giving this opportunity.
Thank you, Dr. Manjunath, for your well-explained product specifications and what we offer as a product for E. coli HCP detections. We have one question from Dr. Akash on the specific details about the regulated market specific guidelines for the E. coli HCP. So would you like to take this?
Yeah, you can go ahead, Dr. Dinesh.
Yeah. Dr. Akash, like when we talk about the regulatory market, one is the regulated and one is semi-regulated. So there is always the regulatory says that any product, because this HCP depends on the product specific as well. Like if you are producing a product erythropoietin or PEG-GCSF or FSH or any other products. So these HCPs will depend varies from product to product as well. And regulatory says that you should address this sufficient safety and efficacy of the product based on the clinical setup. So the standard guideline from the regulators is somewhere around between 1 nanogram to 100 nanogram per mg of the product. That's the HCP accepted guidelines from the regulated market. But even though the regulated market like US FDA or the EMA, they expect now the process specific HCP evaluation and critical assessment and the downstream processing of the product purifications has to be optimized as per that requirement, because it leads to several complications. Like sometime some HCP proteins are interacting with the finished product itself. So how do we address such things? So that's where sometime the regulators recommend to go for LCMS kind of techniques, where you can identify the particular niche HCP which is coming with your product. And to address such regulatory challenges, a process-specific development is recommended.
Yeah, thank you, Dr. Saini. My concern is do we follow the ICH guidelines, or as per product to product it will vary? But what is the central guideline like for HCPs and all? Like you shared about the limits from 1 nanogram to above and all. So are these explicitly mentioned anywhere, or what is the standard that we refer in this case? ICH, EMA, US FDA. So I was asking about that. Where do we derive these upper and lower limits?
Yeah, so upper and lower limits as such, like say considering US FDA or whether it's EMA or ICH, because this, as I said, this level of HCP acceptance depends on product to product. Okay, when we present the clinical data and our phases trial data to regulators, that's where they take into the considerations. But considering our product as a concern, like how we have validated, we are validating the product based on the EMA guidelines, where we take care of all the different parameters like the assay robustness, sensitivity, precision, accuracy, intra-batch variations, then stability of the analyte, stability of our product itself or as a whole product, like how the product is stable. But when we talk about the particular regulatory and particular product, yes, the regulators take this evaluation on case-to-case basis, but this is the broad range mentioned. Like it can be anything between this range, given the proper clinical justification that this product is safe and effective with these compositions and this level of HCP.
Okay.
So, Dr.
Thank you, Dr.
Akash, the ICH, WHO, and FDA or EMA guidelines generally are almost close to each other for
Yeah.
their guidelines for validation of product-specific HCPs. So either you can based on your products, you can either of them follow which guidelines are more suitable for the product which you are using currently, and still
Alright.
do a partial validation based on this kit using the kit, and still get into the results following the guidelines.
So these results, like what we get on ELISA readers and all, do these equipment require a CE certification sort of thing? The plate readers or the equipment that we carry out our assays on?
So the readers have the CA certification, that is the electronic component of the certification. If you do require to generate the data on these equipments, you would require an CFR compliance on that.
Yeah.
The 21 CFR
21
compliance. So
CFR.
that should give you the validity of the data on the equipment which you generate.
Right. Sure. Thank you, Doctor.
Thank you, Dr. Akash. Yeah. So any other questions from participants? I would like to mention, like when it comes to the uses of the product, since it's a generic product, considering the different E. coli strains, as mentioned in our presentation also by Dr. Manjunath. So by and large, using these strains, you can easily achieve a good sensitivity and better coverage with these antibodies and with the product what we have developed here, because we have individually tested the reactivity of our antibody against these strain specific lysate, and analyzed the coverage of our antibodies, which gives a very good coverage of more than 80 to 85% coverage we have achieved using our antibodies. And that's how we recommend these products for all the generic uses, applications. It's suitable. And we can, addition to this, if somebody is interested to evaluate this product performance and suitability in their process, we are open to collaborate in a way that where we can support the samples for evaluations, and you can get your own confidence on the product, how the product is performing in your process specific HCP detections. And then further we can take the conversation continue.
Dinesh, what is the host you raised antibody in, goat or rabbit?
We use both goat and rabbit.
So for both capture and detection, you use the same species or different species?
No, we do the combination of host specific antibody, like rabbit versus goat, checking them into the capture and detection. So we use two different species.
So you use two species, Assay.
Yeah. Yeah.
And your coverage of percentage coverage?
Percentage coverage is more than 85%.
85.
Yeah.
Which technique was used?
We have analyzed using this BioRed software image lab.
Not the G.
Not the GE.
Fluorescence one, no.
No.
Yeah, thank you.
Thank you. Thank you very much, Dr. Anand. Yeah. So we'll wait for, is there any further question? on the product or its regulatory requirement or the technical aspect of the product regarding its performance, uses?
We are good right now. Maybe later when we go through the literature, then we can come up with a relevant question or some query regarding that.
Sure. So, Dr. Akash, I would like to understand from you, like, what sort of solutions you are looking for. Is it something on the similar line what we are talking today?
I'm here to understand the regulatory aspects that we commonly deal in biologics.
Okay.
And when we, conduct the review of the BPRs and all, so it should be the user should be able to understand what type and what philosophy, basically the fundamental that goes on inside the detection, as well as the regulatory aspects that we need to capture for understanding the data and for the filing.
Okay. So to support this, maybe I will also share the technical, complete technical details of this product, and you can just go through the complete technical specification of this product, and you may find more information on what you are looking for, and you can also take a assessment of suitability in your process.
Yeah, that would be great.
Yeah. Thank you. Thank you very much, Dr. Akash. So, any other question? Okay, so with this, I take this opportunity, and I would like to extend my thank you for all the participants and giving us this opportunity to present our R&D work and on the finished product what we offer at deNOVO Biolabs. I thank you, all of you, and I just hand over to Dr. Manjunath if you have anything to say.
Yeah, thank you everyone for the opportunity given here. So we would like to address the queries specific to your requirements, whatever you would have, and we will also support you with the samples for the analysis. The kit can be used from by your end, and you can test your actual samples on the kit and get the actual data, how it works for your samples. So thank you again for attending this webinar. Yeah, Dinesh.
Yeah, thank you, Dr. Manjunath. So I would like to extend my thanks to one and all again for joining this webinar. It was a wonderful interaction we had today, and I hope this will continue, and we will be sharing the lot of technical informations to all the participants who have joined today. Thank you very much.
Thank you, sir.